Okay, so this is the continuation of micro lecture 3, and this is part 2. As you can see, we've been describing the external structures going in. We described the glycocalyx and the flagella and the fimbria and pili and cell wall, including in gram-negatives, outer membrane and cell wall, and in the inner cell and plasma membrane, and in the gram-positives, the cell wall and plasma membrane. And now we'll talk about the cytoplasm. That's the soluble fluid inside the cell membrane, which contains a number of different structures and dissolved or soluble proteins. Remember, there are no membrane-bound organelles in bacteria in the cytoplasm, unlike what you have in eukaryotic organisms. The nuclear area is a pre-nucleus, sometimes called the nucleoid. It does not have a nuclear membrane, of course, and you have the in bacteria you have the single nucleus, single molecule or chromosome not molecule but chromosome, DNA of chromosome, which is circular in structure. It's large, it's coiled back and forth in on itself, so it looks like many, but is only actually one circular chromosome. There are ribosomes in the cytoplasm, and as you know, the ribosomes are involved in protein synthesis using messenger RNA that was transcribed from a gene in the DNA. Messenger RNA is transcribed from the DNA, and then it is used at the ribosome in a process called translation to synthesize the protein that it codes for. So you have DNA being transcribed to messenger RNA being translated into protein. That's the sequence of events. The information moves from DNA to messenger RNA by transcription, and from messenger RNA into protein by translation at the ribosome. Now, the thing about the ribosomes is that they are made of two subunits, a smaller one and a larger one, and these have a characteristic sedimentation rate. They will sediment down in a high density fluid at a certain characteristic rate of sedimentation. These 30 and 50 are not size in terms of weight. It is how it characteristic sediments down through a what's called a density gradient, a solution in a test tube that gets spun in a centrifuge. So these so you don't add them to get this smaller unit is 30S, the larger one is 50, and together they have a 70S. So the ribosomes in bacteria are 70S. Okay, some, not all, but some very interesting ones and important ones in terms of the fact that they are pathogenic. Several of them are pathogenic. Several bacteria species can form what are called spores or endospores. A spore is essentially is the cell at rest or in a state of suspension. It is not growing. The spore forms whenever the bacterial cell, at least the ones that can produce them, whenever the bacterial cell is in harsh conditions that are not conducive to growth, maybe high tonicity or low tonicity, or dried, desiccated, or conditions are too hot, or there's not enough nutrients present. They will form spores, and the spore forms around the DNA chromosome, so it protects that genetic information. The rest of the organism, the rest of the bacteria disintegrates away, and you're left with this very resistant essentially like a resting cell in spore form. Now, there are two important genera. Genera is the plural for genus. So there are two important genera that can produce spores. One is called Bacillus, and the other one is called Clostridium. And what I've listed down below here as examples are species of Bacillus and species of Tetanus, most except for one, that cause diseases, and all of these can form spores. So Bacillus anthracis can cause the disease anthrax. Bacillus subtilis cannot cause any disease. It's non-pathogenic, and that's the one we typically use in the lab when we're looking at spore formation or staining for presence of spores. Clostridium tetani causes tetanus. Clostridium difficile causes diarrhea and colitis. Clostridium botulinum will cause botulism, a very acute type of food poisoning, and Clostridium perfringens causes gas gangrene. All of the Clostridia, I'll repeat this later when I get into more detail on them, are all anaerobic organisms. They grow in the absence of oxygen. Okay, so sporulation is the process of endospore formation, and germination is when they go back to a vegetative state, actually growing. So germination is usually used, maybe you're more familiar with it when you're talking about plant seeds. You know, you plant them in the ground and they germinate, the plant grows. Well, in this case, the spores, once they're in a surrounding conditions that are conducive, that are proper for growth, they will germinate. You get the breaking open of the spore and a bacterial organism comes out, and it can start growing in its vegetative state. And here you see an organism with a spore forming in the middle. Okay, so this means that this one has just formed the spore because it still has the bacterial cell wall around it. Remember, when spores form, typically what happens is once the spore is formed inside the rest of the vegetative part of the cell, the rest of the bacterial cell disintegrates away. These are photomicrographs at high power. The other two figures here and below that are high power photomicrographs of bacterial species that can form spores and have been stained using an endospore stain. Okay, and in this endospore stain, the green stained object is the endospore inside, in this case, in many of these, inside the bacteria, and the red part or pink part is the vegetative cell around it. Remember that it's shown better here that these endospores form inside each of the bacteria, and then the rest of the bacteria disintegrates away, and you see some free, some spores down here that are free without any red vegetative cell around them. I don't know how many you see here. Well, here's one right there. There was one. I'm going to get rid of the highlighting and you'll see it here and here and here and here. There are many examples in the lower one versus the upper. Okay, endospores, Clostridium and Bacillus. Good. So let's move on to eukaryotic cells. Remember, eukaryotic cells, eukarya, the domain eukarya covers everything other than the prokaryotes, and that includes the protists or protozoa that are single-celled eukaryotic cells that are neither plant nor animal. The fungi, the what else? Algae, all the animals, and all the plants. These eukaryotic cells, of course, have a true nucleus. They have a number of different cytoplasmic organelles that are membrane bound. They have many characteristics that are uniquely different from bacteria. And you studied these, so it's really a review. You studied them last year and probably in other places. This is a diagram of a sort of a generic cell, showing a cell. On the right side of the figure is an animal cell, and on the left side, in the left half, is a plant cell. I know that because I can recognize it from the internal organelles that are shown. On the left side, you see what are obviously here, what is a chloroplast right there, and there's another one there. Only plants or algae could possibly possess chloroplasts. Let's move on. Well, actually, no, I'm going to just review some of the organelles. We'll go into them in detail in the next few slides, but I just want to remind you that what we have looking here on the right-hand side of the animal cell, what you have is here a nucleolus. This, of course, is the nuclear membrane surrounding the nucleus that contains DNA. So that's a nuclear membrane, and there in the middle was a nucleolus. Just immediately outside of that and connected through the nuclear membrane, you can see if you look carefully the connections, is the endoplasmic reticulum, which has dotted, and that's all shown all for those of you who don't recognize it. It's all of these flattened, elongated membrane bound sac-like structures. All over it are small dots, and those are ribosomes that are involved in translation of messenger RNA into protein. You also have mitochondria. There's mitochondria, and there's mitochondria. Of course, plants actually do also have mitochondria, but we don't study plants here. We study animal cells, eukaryotic cells. Outside, further out from the endoplasmic reticulum shown in green, you have the Golgi apparatus. Again, a series of membrane-bound sacs, and it's shown in light blue in this diagram. You have here also shown some membrane-bound lysosomes and peroxisomes, and I see a centriole here in this area. And of course you see a flagella. There is, of course, in the human only one flagellated cell, and that is the sperm in males. Other than that, there are other organisms that have flagella, particularly protozoa or protista, that swim around in water. And of course the whole thing is surrounded by a cell membrane, a plasma membrane. Okay, some of the ones I would expect you, or structures I would expect you to know, and that we've covered certainly in the past. We're looking at a eukaryotic cell, and pretty much all of these structures go for any eukaryotic cell, not just animal cells, but any at least protista, protozoa. Also, many of them have flagella. There's a nucleus, nucleolus, Golgi apparatus, sometimes called the Golgi complex, the cytoplasm, lysosome, ribosomes, microtubules that act as insoluble framework proteins, sort of cytoskeleton. I'm reminding you of things we covered in the past in anatomy and physiology last year. The rough endoplasmic reticulum, mitochondria, smooth endoplasmic reticulum, and plasma membrane. All of that is review. Here you see a protozoa, a eukaryotic cell with that's neither plant nor animal, having a flagellum. This one has cilia. Flagella and cilia in these kind of eukaryotic organisms are used for motility. The flagella and the cilia have a typical arrangement of along their axis. This is a cross section through a flagella. and you see the arrangement of these long microtubules. It's typically 9 doublets and 2 central microtubules. So one, if I remember correctly, it's 9. 1, 2, 3, 4, 5, 6, 7, 8, 9. Yes. Okay. And they run the entire length of the flagella, and they cause it to allow it to motion so that the organism can move through an aqueous solution. The cell wall is different in different kinds of organisms. Animal cells do not have a cell wall. They do have a cell membrane. All cells have a cell membrane, but animal cells do not have a cell wall. But plants do, algae does, fungi do. They all have cell wall. And there are many different carbohydrates in that cell wall. Okay, the plasma membrane is very similar to the one in the bacteria, very similar to all cells. In fact, the cell membrane or plasma membrane is a phospholipid bilayer, a single layer of phospholipid a single membrane phospholipid bilayer with both peripheral proteins and integral proteins. And some of the integral proteins are in fact transmembrane, as we spoke about before with the bacteria. And we're talking about a selectively permeable cell membrane or plasma membrane that doesn't allow the passage of anything. It allows some molecules to pass through, and others have to pass through with the help of proteins that are in the membrane, and others have to be moved through by active energy requiring active transport processes. Some processes of movement across are not limited. They're not selectively permeable. They just let the molecules move across, and that is simple diffusion for solutes and osmosis for the solvent in living systems, water. So the movement of water across a semi-permeable membrane is in fact osmosis. Now some things can move across the membrane and into a cell by endocytosis. And endocytosis, a form of endocytosis, moving insoluble things across into the cell, is one process is phagocytosis. The main phagocytic cells in humans are neutrophils and macrophages. And the process involves extension of what are called pseudopods. The cell will extend areas of its cell membrane, engulf around a particle or bacteria or anything foreign, and then encircle it and fold it into itself, into what's called a phagocytic vacuole or a phagosome. Okay. Another type of endocytosis is penocytosis, and there it brings in fluid or dissolved substances by small folding in of small folds of the membrane around the fluid and bringing it into the cell. Eukaryotic cells, of course, have a cytoplasm. They have a cytoskeleton. You learned about this. They are insoluble fibrous proteins that make up microfilaments, intermediate filaments, and microtubules. Now, the cytoplasm is not static. It's not sitting there like in a frozen state. There is streaming. There's even movement of the organelles through the cytoplasm as the organism lives and goes through its life cycle, or the cell lives and goes through its life cycle. There are many characteristic membrane-bound organelles, and when I say membrane, it's the same idea that it's a phospholipid membrane. The nucleus is bound by a nuclear membrane, surrounded by a nuclear membrane. One thing that you may not have learned about the nucleus is that the nuclear membrane around it is a double layer. It is two phospholipid membranes. There is also, of course, the endoplasmic reticulum, and then the Golgi apparatus or Golgi complex, and lysosomes and vacuoles and mitochondria and chloroplasts in plants or algae. I've listed the functions, so you can read it on your own. This is a review for you. Okay, let's just look at these different parts of the cell. Here you see a diagram showing this is the same diagram actually that we looked at originally. And here you see in color the nucleus with the nuclear membrane, and in the middle of the nucleolus it has a phospholipid bilayer, two membranes here. It's a double membrane bound organelle or structure. So you have a double membrane here and a double membrane right beside it there. There are pores, and through these pores things can move out, and they can move out into the endoplasmic reticulum through those pores. A typical example of something moving from the nucleus into the endoplasmic reticulum would of course be messenger RNA coding for different proteins following transcription of genes in the DNA. And here you see the endoplasmic reticulum suspended in this form outside of the nucleus, outside of the nuclear membrane, in these flattened membrane-bound sacs. You have some areas that are smooth without ribosomes and rough, those have ribosomes. And the small dots that you see here in this electron micrograph, that's a photograph taken through use of an electron microscope. What you see are those small dark dots, and each of those is a ribosome. Ribosomes in bacteria, I mentioned, have a sedimentation rate of 70. Okay, there's a small subunit and a larger subunit. The small subunit in bacteria, I'm talking about bacteria now, is 30 and 50 S. You don't add them up when they are combined together. The two parts together forming the ribosome have a sedimentation rate of 70 S. Okay, and as I've told you before, I mentioned to you before, chloroplasts and mitochondria both have the same kind of sedimentation rate. They have, for their ribosomes, they have 70S ribosomes, just very similar to the ones in bacteria. In eukaryotic, remember, I remind you that these organelles have their own DNA. Okay, let's not talk about chloroplasts, but the same goes for chloroplasts as mitochondria. The mitochondria have their own DNA that codes for them. They are self-replicating organelles that code for their own synthesis, and therefore have their own DNA, have their own genes, making their own proteins, and therefore they have the ribosomes themselves inside them. The cell has ribosomes, eukaryotic cell have ribosomes attached to the endoplasmic reticulum, and that would be rough endoplasmic reticulum, and some ribosomes that are free-floating in the cytoplasm, and these are 80S in their characteristic sedimentation rate. This is the Golgi apparatus that's involved in modifications of proteins that were made in the endoplasmic reticulum and were transported over to the Golgi apparatus in these transport vesicles. And then the transport vesicles fuse with the membrane of the Golgi apparatus, and the newly synthesized proteins made in the rough endoplasmic reticulum are modified, changed in the Golgi apparatus, and then they are, if they're going to be secreted out of the cell, they will go to the surface of the cell in these secretory vacuoles that fuse with the cell membrane and release them to the outside. Okay, where are we? 54 or 59, very good. So, again, except with an electron microscope as opposed to a drawing, you have here a drawing of a sort of a photomicrograph of a sort of characteristic cell. On the upper left is a plant cell, and on the lower right is half of a animal cell. And the animal cell in this case is a plasma cell, and I remind you that plasma cells are B lymphocytes that have been activated and B lymphocytes that get activated, they're activated because they've come into contact with their specific target antigen. Okay, they have a antigen receptor on their surface, antibody molecule on their surface, specific for a particular antigen, and if it gets activated, it's because it's contacted and bound its antigen, and then it changes from a B lymphocyte into a plasma cell, which starts synthesizing large amounts of antibody molecules and secreting that antibody into the surrounding plasma or interstitial fluid. So this is a plasma cell, and plasma cells are very busy making antibody protein, so not surprisingly, and when you look here, it's full, literally full of rough endoplasmic reticulum. All of that I just highlighted is rough endoplasmic reticulum. Okay. So, did I miss a slide? I don't think so. So, another membrane-bound organelle in animal cells, in eukaryotic cells, are the mitochondria, also in plants, but in animal eukaryotic organisms are mitochondria. Remember, I told you that the mitochondria have a 70S ribosome, just like bacteria. They also have an outer membrane, just like gram-negative bacteria. They have two membranes, an outer membrane and an inner membrane. Phospholipid bilayer inner membrane and phospholipid bilayer outer membrane. So that's another characteristic that is similar between mitochondria and bacteria. The area between, or the area on the area on the inside of the inner membrane is called the matrix. Okay, and the chloroplasts are similar in that they have an inner, and they have, well, you know what? Let's not talk about chloroplasts. Let's move on. So we've discussed several things that evidence that does, and characteristic structures that are common between mitochondria and bacteria, and a few of the things that point to the fact that mitochondria in fact are evolved from bacteria, that at some point during evolution started living in a symbiotic relationship inside another cell and developed into eventually a stable relationship and developed into the eukarya. And that happens to be called the endosymbiotic theory, or endosymbiotic model for development of eukarya in this manner. And so we come to the end of Lecture 3, Part 2.